This Week in Virology, the podcast about viruses, the kind that make you sick.
From Microbe TV, this is TWiV, This Week in Virology, episode 869, recorded on February 18th, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, Vincent, and everybody else. Well, today out the window, it's absolutely gorgeous. There's puffy clouds all over the place. The temperature is about 55 degrees, perhaps a little bit less than that now, and falling. We expect this to reach 30 degrees by the time the evening time arises, but heck of a day out there so far. Typical a spring day, not a winter day, but we're not there yet.
It's 4 degrees Celsius here.
4? Oh, okay. Maybe
it's 4.
I was giving you an earlier reading than, I guess, around 10 or 11 o'clock. Okay.
Yeah, there was a lot warmer, but it's getting cold now.
Yeah, it's going to come through.
Also joining us from Madison, New Jersey, Brianne Barker.
Hi, great to be here. It is 41 here. So it has definitely cooled down quite a bit, although it is still bright and sunny. And we have had tremendous wind over the past 12 hours or so.
Yes, we have. Yes, we have.
Such that I'm kind of surprised that I'm here and not in Oz.
Oz. Yeah. Also joining us from Austin, Texas, Rich Condit.
Hi, everybody. We got 56 degrees. So for me, that's a little chilly. It's sunny. It came to near freezing last night. It's going to go to near freezing tonight. And Brianne, you have the wind today that we had yesterday and the day before.
Oh, interesting.
So working its way across the country.
The annual Biomedical Research Conference for Minority Students is launching a new meeting, the ABR-CMS e-Poster Spring Symposium for Emerging Scientists. For selected high school students who've conducted formalized research, community college students, and first-year undergrads to present research in one of the 12 ABRCAMs, right? Is that what you say,
right.
Brianne? ABRCAMs? One of the 12 ABRCAMs disciplines. This meeting held online Saturday, April 23rd, will help students prepare and qualify for subsequent ABRCAMs participation in the annual forum meeting. Abstracts due March 15th. In addition, ABRCAMS wants to promote professional development of early career scientists by encouraging grad students and postdocs to sign up to be abstract reviewers and judges, although all active researchers are eligible to volunteer. So ABRCAMS 2022, November 9th through 12th, Anaheim, California. In addition, ASV registration is now open. Abstracts are due March 1st. Travel grant applications for students, postdocs, and teachers of undergraduate virology are due March 3rd. Applications for ASV CARES grants are available for members to support dependent care at home or elsewhere during the meeting. On-site daycare will be available for members. Details to be announced very soon, similar to what we planned for the 2020 ASV meeting at Colorado State. For everything, go to asv.org and follow links to the ASV 2022 meeting. And the Department of Microbiology and Immunology at Louisiana State University Health in Shreveport would like to encourage people interested in pursuing graduate studies in fall 2022 to apply to our MS and PhD programs. We are a vibrant department of 16 faculty members working on a variety of DNA and RNA viruses, innate immunology, and intracellular bacteria such as Bordetella and Legionella, located in northwestern Louisiana. We offer unique opportunities to grow personally and professionally, milder winters, and southern hospitality. The deadline for applications has been extended to March 1st. Check the department website. There'll be a link in the show notes. All right. Rich, I have to apologize up front. I'm sorry. Rich sent me a note this week. He said, thank you for doing one paper the last few weeks. It's really good. We can focus, and today we have 3, but I assure you, the 2 of them will be brief.
Yeah, it's okay because in particular the last 2, they're so interdependent. It would be a shame not to do.
I have to say
both
that
of them.
I've seen them both on Twitter,
right,
and had made a note that I really wanted to read them, but hadn't had a chance to do that, and was so excited when I saw them in the show notes that I was finally going to get a chance to read
great
them.
Papers. However, we're going to start with a very short look at a paper which was published in 1954.
Right.
And this is the discovery of viremia in poliomyelitis by Dorothy Horstman. Remember was it Friday? We talked about... maybe it was Tuesday. I don't remember.
Who knows?
Echo viruses, and her little quip at the bottom of an article saying, what an echo virus is. And I mentioned she discovered the viremia. And I said, we should take a look at that paper because let's see what they did and compare it to what they would do today. So this is the Journal of Experimental Medicine. And you wouldn't know it because the name of the journal isn't anywhere on the PDF that I have, but you can tell from the formatting.
That was published out of the Rockefeller University, wasn't it?
Yeah, that's right. And so this was received for publication January 1954. Viremia in human poliomyelitis. Dorothy Horstman is the first author, Robert McCollum and Anne Mascola. It's Yale University School of Medicine. I was 1 year old at the time. Just 1 year old, 1954. So they give you some nice background here, and they say the search for virus in the blood of patients with polio has been looked at since 1909. Negative results, and you can't blame them. They didn't have cell cultures until the 40s and 50s.
Or animal models.
Well, they had animal models, but that's hard, as you'll see here. 1946, one isolation of virus from blood by the Horstman lab in 1949. A second, Hillary Kaprowski apparently did that. And they say the positive in our lab was from a 9-year-old girl who it was drawn within 6 hours from the onset of minor illness. Now, when you get infected with poliovirus, most of the people get nothing, subclinical illness, or a minor illness, headache, fever, that sort of thing, and you recover. And only 1% proceed to paralytic disease. And so this, they say, the blood was drawn from this girl within 6 hours from the onset of some minor disease. And it was probably during an outbreak where a lot of kids were being infected, and so they could see that this might be a poliovirus. And they said...
Vincent, set a little context for me here, because my sense from looking over this paper is that what you just described, that there's only, say, 1% that progressed to paralytic disease, and the succession of events, they were just beginning to understand when this was done. Is that a fair statement? How much of that sort of pathogenesis was understood?
I think the rarity of the paralysis was understood.
Okay.
I think the minor illness was known, but how they were connected wasn't quite understood.
Okay.
By that time, though,
how you get from one to another.
Yeah.
The town that I moved to from California to New Jersey, Dumont, had a swimming pool. And when we first moved there, of course, the swimming pool was open. And by the time that paper came out, they had closed the swimming pool because they had discovered the epidemiology portion of this.
Yeah, that's what they often did that in the summer. They would close pools because kids would hang out and they'd transmit it,
Exactly.
Anyway, she says this one child positive was 1 out of 111. So they said, oh, it's rare. Viremia is rare and no significance to the pathogenesis. I think this is really interesting because it illustrates how you make missteps, right? But then they realize that by the time you see paralysis, the virus is gone, and you have antibodies, actually. And so that may make it hard to isolate virus from the blood. And they have they had some primate evidence for that. You had to use primates for for studying non-human primates. There were no other animal models. So she says, I think we've been looking too late, because if you look after paralysis, the virus is gone. So there are two phases. There's this early minor illness, which in most people goes away. And then there's the second phase, which 1% they can get paralysis.
And Vincent, that early minor illness, is that primarily respiratory, primarily GI, little of everything?
Actually, it's neither. It's the virus, you ingest it, it goes into your intestines, it reproduces. I think it's a general inflammatory reaction, right?
Okay.
Because there's no GI pathology whatsoever. There's no diarrhea, gastroenteritis. There's no pathology in the GI tract.
And there's no respiratory pathology either. This is a gastrointestinal thing.
Yeah, there can be fever. Yeah.
Right?
Fever is one of them.
Malaise, something like that.
Malaise, the old flu-like illness, right?
Yeah.
Right.
It could be caused by
a stomach virus.
Yeah, exactly. So that's what they did in this paper. They had an outbreak of polio in Ohio, in rural Ohio, and they used this to get specimens. This was June and July 1952. They got blood, they got throat swab, rectal swab, and of course, the rectal swab, they would look for virus, and the blood, they would look for virus, and the throat swab, they could look for virus, and the blood, they would look for virus and antibodies, of course. No PCR in 1954, folks, which is probably a good thing. And here's how they looked for virus. Rhesus monkeys, right? They take a little bit of the centrifuged blood and they inject it into the monkeys. And then they have cell cultures of various sorts. One of them is monkey testicular cells, and they have roller tubes. Rich, you know what a roller tube is.
I do indeed.
And they have, they look for cytopathic effect, right? You put the blood or whatever serum on and you look for the cells rounding up, because how else? There's no PCR to measure it and so forth.
No plaque assays?
So 1954, they actually should, because it was developed in 51 by Delbecco. Yeah, they should have plaque assays, yeah. But they don't report them here, no.
That was new technology.
And which cells were they using for their plaque assays? Fibroblasts?
Didn't Delbecco use HeLAs?
HeLa, that's right.
I don't know.
I'm not sure. I'll look it up.
And
it would be early.
Was it on that stuff?
Because the HeLAs were made in 51, right?
And Weller was another author on that.
So
paper.
Enders, Weller, and Robbins, what they did was to take fetal tissue that was, being discarded, and they made cell cultures, and they showed that the virus could grow in that, right? Which was important because everyone thought it was strictly neurotropic, right? So anyway, they do isolate virus from 6 of 33 individuals, which is more than they ever had before. And most of these kids recovered, I believe, and they just think that they got it early. But what's interesting is that all of the samples put in monkeys gave nothing. They had to pass it in cell culture multiple times to get enough virus, and then when they put that in monkeys, it would paralyze them, because there's not a lot of virus in the blood, apparently. So insufficient amount of virus to produce polio in rhesus monkeys on direct intracerebral inoculation of undiluted plasma.
So, Vincent, is that true for people, that the dose matters as to whether you're going to go on to get paralyzed?
I don't know of any evidence about that, because who knows how much you get, right?
Well, yeah, because the virus obviously can replicate once it's inside and achieve higher numbers.
Well,
right?
if you put small amounts in, it's not successful at initiating an infection. So it's sort of like the human challenge study. I don't know. Were you on TWiV when we did that? I think so. The SARS-CoV-2 challenge study.
Oh yeah. Okay.
Yeah, they gave them 55 TC-ID50s, and half of them got infected. So it's not enough to infect everyone.
Right.
So it's fresh.
So, but also, what do they measure in the monkeys?
Polio.
They measured
paralysis.
Polio.
Yeah.
So the idea is that at some point, some of those monkeys were not getting infected, and some fraction of the monkeys may have been infected but not progressed to polio.
Just
Probably.
Like just like
Probably.
people.
Yeah.
And the ones that get so they develop an immunity. Then you would expect that to result in an immunity, right? The ones that didn't get any symptoms,
the monkeys?
still got people that still got infected but no symptoms.
Oh yeah, they develop immunity within 7 to 14 days. Yeah, they get. You can see antibodies.
Then they don't never get infected again. Then is that right?
Well, there are 3 serotypes. Remember?
Oh, that's right. That's right.
And so
sure,
you can get infected with a different serotype.
Sure, absolutely.
Yeah, and it looks like they're listing these kids as having, headache, anorexia, vomiting, as their symptoms. So random flu-like symptoms.
Timeout. I have to interject something here.
Yeah.
I found a little line here that by the 1950s, techniques for reliably producing and propagating cell cultures were developed. And in 1951, the first HeLOs were isolated. Delbecco took advantage of these advances and showed in 1952 that Western equine encephalitis virus formed plaques on monolayers of chicken embryo fibroblasts.
Cool.
There you go.
That's from an obituary for Renato Delbecco that appears on this. It's called Virology blog.
Did I write that?
I guess you did.
Okay, that's what I used to write. Yeah, and the thing is, the chicken embryo fibroblasts. Then Howard Temin came to his lab to do a postdoc, and he took them and he infected them with Rous sarcoma virus, which up to that time people had only been able to work with in chickens, and you can't do very much. And he showed they made foci in the chick embryo fibroblasts, and that was the road towards identifying what was going on.
Chickens were used to identify Rous sarcoma virus.
Yes, but you can't do much biochemistry in chickens.
No. But they sure taste good.
Back then. Anyway, they say here, kidney tissue tubes, it's funny, they call them tissue tubes, proved more sensitive in detecting virus than testicular tissue tubes. And now, in fact, the virus is propagated in monkey kidney cultures for vaccine production to this day. So
Rhesus or vervet?
Rhesus or vervet?
Vervet, yeah.
Vervet, probably vervet. And I wonder if the problem is that you can't do a plaque assay in a tube.
Very easily.
Brian, you remember they used to have glass flasks, right?
Yeah.
Now, today they have plastic flasks for cell cultures, but they had glass with stoppers in them. I don't know how they kept these things from getting contaminated. Stoppers. And you could do plaque assays in those. And I've seen pictures of them in glass flasks.
There was no such thing as a biological safety cabinet.
And we used to rewash them.
So everything's done out on the bench. You just sort of brush the dust bunnies aside. And
when I joined Palazzi's lab, we didn't have any cell culture hoods. We had a room with a stainless steel top, and it was closed. And when you left, you turned there were UV lights on the ceiling. You would turn those on. So every time you go in to work, you got that smell of ozone, right?
Yeah.
Yeah, we didn't have a tissue culture hood.
So you had a tissue culture room where you also spent time.
Yeah, it was a room, and there would sometimes be 2 people in there, working on 2 benches, and I didn't think twice about it. It seemed. When did I encounter a hood?
Any laboratory transmitted polio?
Yeah, there have been. Anyway, so that's the story here. I wanted to illustrate how, you don't always understand what's going on, and you have to figure it out, and sometimes you make mistakes.
How soon afterwards was the vaccines brought out?
A year later, 1955.
Look at that.
So Salk was working on them, yeah.
So back to the earlier discussion about the sort of the time course of pathogenesis. Okay, so they now here in this paper make an association between early mild disease and viremia.
Yeah, right.
And I think they if I remember correctly, they're tentative about the relationship between that and paralytic disease. Mm-hmm. So what's the answer? How does the virus gain access to the nervous system? Through the blood?
Yes, that's the idea, but and there are two possibilities, right? There is virus crossing capillaries in the brain, which does happen. People have shown that. Or entering a muscle and being transported axonally to the brain from the neuromuscular junction. That happens also, probably two ways.
Not from the gut tract through the myenteric plexus?
People have tossed that about, but I don't see evidence for it, no.
Okay.
Yeah.
Because, I think that's how some other agents are transmitted through via that.
Absolutely. So, for example, reovirus, which you can study in mice, Bernie Fields showed if you cut the vagus nerve, which goes right from the gut to the spinal to the brain, and you feed mice reovirus, it no longer makes it into the brain. So it's a very nice experiment.
And so the paralysis is a result of pathogenesis in the brain rather than in the peripheral nervous system, correct?
That's correct. It's in the central nervous system, brain and spinal cord, yeah. So they say here in the discussion, we've found virus in the blood in 3 types of infection: minor illness, asymptomatic, and just before the non-paralytic form of the disease, which is, some of these kids would get headaches or neck stiffness, right? That was considered non-paralytic. It was clearly involving some nervous aspect. And they say, how commonly viremia occurs, one cannot say. I love one cannot say. But since we found it in 5 of 14 individuals, it may occur with some frequency. And then later on, it was found in many people. So it was quite clear that it was a part of the pathogenesis. But, there was a big discussion here. For example, the relationship of viremia to central nervous system invasion is uncertain. Some people thought it was a spillover, that a virus really was strictly neurotropic. It only grew in the brain and spinal cord and it spilled over into the blood, and that's that was.
Oh, dear.
But she says Bodian did experiments where he could protect monkeys from paralysis by giving them small amounts of antibody. They would orally infect the monkeys, inject some antibody, and that protected them. So they think that the virus in the blood is actually vital to the pathogenesis, and that's what ended up being the case. That this is not a strictly neurotropic virus. It reproduces in the intestinal tissues and then makes its way in the blood. And they suggest a time course of events. First, primary multiplication a few days after exposure. Such multiplication may be largely or entirely extraneural, like gut. Second, the development of the minor illness in association with virus in the blood, throat, and feces. Viremia persists only a few days, and then antibodies come up. And finally, circulating virus may enter the CNS and result in either, minor paralysis, not minor paralysis, what do they call it? Minor symptoms or full-blown paralysis.
So the virus thinks it goes through the choroid plexus, maybe?
I think it's possible, yeah. Choroid plexus, as you know, is the source of cerebrospinal fluid.
Yep.
It is also the most permeable part of the CNS. The capillaries are very permeable in the choroid plexus, so it's a good place for virus to get in, yeah.
So, the paralytic disease is only a small fraction, like 1% of the infections. Do you have any feel for what percentage of infections result in a viremia?
Oh, I think they're all
viremic?
It's just a matter of whether you detect it or not. Yeah.
Okay.
Yeah, I think they're all viremic. Yep. And I just wanted to end with this last paragraph. They're talking about antibody. And they have some evidence that you could, if you add antibody to a virus, you could actually dissociate it by diluting our virus antibody mixture. And they say, this is very funny, since the nature of the union of poliovirus and its antibody is unknown, One can only speculate as to the intimacy of the combination and the difficulty or ease with which it can be separated. It's so funny, isn't it? I
really like that. We've talked about this before in these older papers, the sort of conversational style.
Yeah,
it's exactly really nice.
Right. And so the other thing I thought that was funny, in the way these new JEM papers used to be written, they'd have big text, and all of a sudden the text would get small, where they're talking about kind of peripheral stuff. And I thought, it's really supplementary data, but it's right in the text, right?
That's right. And
supplementary font.
At that point, who was the editor-in-chief for the Journal of Experimental Medicine? Because at one point, Peyton Rouse was the chief reviewer and in charge of appointing their editorial board, etc. He was complete control.
I don't know who was in 1954. But he was probably finished by then because he got his Nobel in 65, right?
Well, he couldn't have been finished that long ago because I was there when he was still there.
What years were you there?
I was there from 67 to 70.
And was he active still?
Well, he was coming in every day and he was ruling the roost, and then he got pancreatic cancer and he died.
He probably came in to have lunch and then went home.
No, he came in to edit papers and to select the ones that agreed with his hypothesis.
You ever hear the poem by John Updike, V.B. Wigglesworth?
No. He was a medical entomologist.
Who? John Updike?
Wigglesworth.
Anyway, it's a fabulous poem where the guy comes to the lab by noon and he does all this stuff, lines and lines of wonderful prose, and then he goes, and by 1 o'clock was gone.
He wiggled his way out of a whole day of work.
It's so fun. He does all these experiments. He calls the FRS. He has the sperm club in for tea. That's one of the lines. It's just great. And then, then you read the last line, and by 1 o'clock he was gone. It's like, what? 1 p.m., 1 a.m., when?
Wigglesworth worked on triatomid bugs.
Which day?
Reduvidae. He worked on reduvidae.
Oh, reduvid bugs? All right. Anyway, I thought that would be a little fun diversion.
Yeah.
We don't do old papers very much.
No, this was great. I loved it.
All right. Then we have two papers which... Sorry, go ahead.
Oh, I was just going to say, old papers are always great to sort of actually learn where the things came from.
No question.
Yeah.
So I have the cover page of the Journal of Experimental Medicine from 1954, volume 99.
Yes.
It says, edited by Peyton Rouse, René Dubois, Herbert Gasser, and Vincent Doyle.
My God.
Wow. I know the first two.
Yep.
Cool. Did you actually talk to Rouse Dixon?
Yeah, I did. He was a mellow old man by then because he had already been accepted into the Connecticut Society for Medical. The AMA branch in Connecticut finally accepted the fact that viruses could cause cancer, and then they reluctantly let him in. But he was a strident individual. He was very difficult. But by then he had said made his. This is after I believe he received the Nobel Prize for that too, didn't he?
65.
So he thought of becoming a member of the Connecticut AMA Society was higher up than getting a Nobel Prize for this thing.
That's what I was just going to ask, what the relative years were. So, it's one thing to get a Nobel Prize, but it's another thing to have Connecticut accept you.
Right.
I think so. I am currently reading Paul Offit's book, Vaccine.
Mm-hmm.
And it tells the Rouse story. And It tells about the chicken farmer coming to Rouse's lab with this chicken with a tumor in it, saying, "What's up with this chicken?"
Indeed.
That was the beginning of the whole thing. If I'm not mistaken, Rouse didn't get his Nobel Prize till he was 85 years old.
That's correct.
Right.
Yeah. Rich, I saw the slide.
Oh, cool.
Somebody there actually said, "Would you like to see the slide?" And I said, "Which slide is that?" And he said, "The slide."
What do you mean, which slide?
It was in one of those histology cabinets, with all the slides that you pull these big racks out on.
Yeah.
And he reaches way in the back and he brings out the slide and he puts it under the microscope. And there it was. There was the tumor.
Cool.
So they must still have this slide, right?
They do. How exciting was that, though? That was, when you think back on that experience, not right away, but over time, like since I became part of this group, and here I am involved in viruses, and I don't know anything about them, and yet I've got connections that go right back to the very beginning.
Oh, I wouldn't say you don't know anything about viruses.
Well, not as much as you guys. I can't.
I mean, you have assimilated something from being on TWiV for what? How many years?
Yeah,
2008.
I hope you can't peer into my brain to find out how much actually stuck.
14 years this fall.
I love the banter that we established. This paper elicited a lot of sidetracks, which filled in a lot of history.
That's what I think we do. I feel we just start talking and we all banter, right?
Yeah, no, that's right. And you want another cup of coffee. You feel like you're sitting around having a clutch.
Was the original goal to capture our banter. And I don't know if banter does it justice, because banter sounds, not useful, right?
Well, we're going off the top of our head for some of this stuff, and we're asking fundamental questions. When Rich, you asked if it was a central nervous system thing or not, or a peripheral nervous system thing, I said, well, don't you remember everybody if they had paralysis, they went into an iron lung. So why did they do that? Because their diaphragm was not being stimulated by the cerebrum.
Well, I was in high school from 1962 until 1966. And there was a couple of kids, on crutches and leg braces.
That's right.
Okay? And you could, that was all because they'd had polio 5 or 10 years earlier.
This violin player, Yitzhak Perlman, and all kinds of other people that have really had remarkable careers, and they didn't let it stand in their way.
Neil Young.
Neil Young, I didn't know that.
Remember?
I think they said both Neil Young and Joni Mitchell.
Yes.
Oh, Joni Mitchell also.
Yes,
wow.
Joni Mitchell also.
That's right. So
Go ahead, Rich.
Dixon talking about the Peyton Rouse's slide reminds me of a couple of stories. We probably all have these of encounters that we had early in our scientific career when we were young, naive punks and didn't understand what was going on.
Right.
That later on you look back and you say, holy cow, that really happened. I'll save this one.
No, those editors that you mentioned, Rich, I became a good friend of René Dubose, a really good friend. I mean, we sat down at the bar at the Rockefeller, and we had lots of extended philosophical conversations about society. And he was writing a book called So Human and Animal at the time, and it turned out to be a real bestseller. It was a Pulitzer Prize winning. Well, he was a polymath. He had knowledge in lots of different directions, but he was one of the greats. And
Brianne, he was like a polyfunctional T cell.
Indeed.
Excellent.
A natural killer.
I was at a meeting at Scripps many years ago, and they had Yitzhak Perlman play for the audience.
Oh, great.
At the end of the meeting, and he came out on his little wheelchair. It's not a wheelchair, but it's a little wheeling device that he pushes himself on. And he talked a little bit about having polio, because I talked at the meeting about polio, and it was about viruses, basically. He talked a little bit about that. I hadn't known before that he had polio as a kid. He can't walk. All right, so now we have two papers which have made the press, and they're all about Epstein-Barr virus and multiple sclerosis. The first is an epidemiological paper, and the second is a mechanistic paper. It's a great pair.
So, Vincent, before you go any further,
Yes.
Epstein-Barr virus has been implicated in lots of different diseases, right?
Yes.
Could you
it also infects pretty much everybody.
Could you give us a short list of those important diseases? Isn't mononucleosis one of those?
Yep.
Well, it does cause mono for sure. Mm-hmm. Infectious mononucleosis is caused by Epstein-Barr virus. As is nasopharyngeal carcinoma.
Keep going.
And
Burkitt's lymphoma.
I meant to mention that too.
A couple of other lymphomas.
Those are definitely caused by EBV, but the association between EBV and MS has always been suspected but tenuous, because 99% of the population is infected with EBV. How do you ever establish etiology, right?
Exactly.
Right. Well, and that's also the same issue that we see with a lot of autoimmune diseases. With a lot of autoimmune diseases, we understand the immunology of what's going on, keeping your disease going from a day-to-day basis. We can say, oh, look at this T cell destroyed this myelin sheath, or this antibody was involved in destroying the myelin sheath. Yeah. But you can't ever say what made the T cell or the antibody start on its autoreactive path. Right. What initiated the event. And usually the idea is that it was, that was maybe 10 years before you got symptoms or the patient had symptoms, and you can't, go back in time and see what virus did you have 10 years ago or what event happened to you 10 years ago.
So I understand that this virus, maybe others have a similar rate of stealing genes from their hosts, but this virus accumulates lots of genes that they didn't have before. And they incorporate them into their genome, right? And that's where
maybe on an evolutionary scale. Is that a stretch? I think that's a stretch. Okay, what I know of EBV is stable. Okay.
Yeah.
But you raise an important point, Dixon. While we're at it, let's do quickly EBV. Okay.
Thank you.
It's a herpes virus. That means it's a double-stranded DNA virus. Has I'm guessing 100, 150 kb genome. That's a guess. 100, 150 genes, something like that. It has both a capsid and an envelope. Okay. So it's got an icosahedral capsid, but it's also got an envelope around that. The herpes viruses as a group have the family has lots of different familiar human members. And of course, there's a lot of other species involved as well. But the human viruses include not only Epstein-Barr virus, but varicella zoster, that's responsible for both chickenpox and shingles, and cold sores, HSV1, and genital sores, HSV2, roseola, right? What's that? HHV6 or something like that. I'm probably going to mess that up. There's 7 or 8 of these herpes.
Kaposi
sarcoma.
Exactly.
Good, thank you. They all have in common, most or all of them have in common latency as part of their life cycle, so that they can infect you, and then whatever their ultimate target cell is, they can park their genome there for basically an indefinite period of time. So you're carrying around their genetic information forever. And depending on the virus and the target cells, this manifests in different ways. So some of these viruses target nervous tissue. So that includes HSV type 1 and type 2, the cold sores and genital sores, and varicella zoster virus. And the most so you get cold sores. Some people get cold sores repeatedly, and that's because the genome is latent in nervous tissue and can re-erupt now and then. The relationship between chickenpox and shingles is a reemergence of a latent genome from an initial infection that caused chickenpox. EBV is one of a group. Oh, cytomegalovirus. We didn't do cytomegalovirus, and that comes up in this as well. Yeah. EBV is one of a group of herpes viruses that infects lymphoid tissue. Okay, and in particular with EBV, it's B cells. All of these viruses are very widespread in the human population. Okay, with the possible exception of HSV2, which its transmission is limited somewhat by the fact that it's sexually transmitted. You find that 95% of the population is infected. And that includes the cold sore virus, HSV1, and EBV, and cytomegalovirus. You get infected when you're very young and carry this thing around forever. And only a fraction of the people who are infected come up with any symptoms, which is also typical for lots of different viruses, including these. So most people get infected with EBV and a bunch of other herpes viruses and never know it. They carry it around. EBV is transmitted by saliva, by the way. But under some circumstances, symptoms can come up, And as we've already discussed in the case of EBV, those are manifested in a bunch of different ways depending on the circumstances.
So,
Mononucleosis is one, Burkitt's lymphoma is another, nasopharyngeal carcinoma is another. There's a few other B-cell lymphomas associated with EBV. And that's the story.
SSPE also, yes.
No, that's measles.
No, that's measles.
Oh, measles.
That's
Sorry.
measles.
Sorry.
And so MS, multiple sclerosis, is a chronic inflammatory demyelinating disease. The myelin sheath of nerves degrade, not known what causes it, but it's the degradation of myelin is immune mediated. It's been suspected that the trigger is a viral infection, and some evidence that EBV is involved. Increased risk of MS after mono, elevated antibody against the viral nuclear antigens, and the presence of the virus in lesions, the myelinating lesions in some pathological studies, but not really good proof. But I think these two papers are really strong. This first one is longitudinal analysis reveals high prevalence of Epstein-Barr virus associated with multiple sclerosis. So this is by Bjornvik, Cortese, Haley, Kuhl, Michael Minna, Yume Lang, Stephen L. Edge, who does the ViroChip. We did one of his papers before. Niebuhr, Scher, Munger, and Asherio from all different places in Boston.
Boston?
Now, the
wicked.
This is very cool.
This is a wicked cool.
Yeah,
This is what they say. Causality implies that some people who develop MS after infection would not have developed MS if they had not been infected. That's causality, right?
That sounds right.
And they say the gold standard, you can't do a trial, they say. We can't do a randomized trial where we infect some people and not others. No. Is an experiment of nature, they call it. A investigation, a longitudinal investigation of the incidence of multiple sclerosis in a cohort of people who are not infected with EBV. Some of them will get infected, and you can follow them up because 95% of adults get infected. So
they used Army personnel, right?
They used an
this
Army
is amazing.
database.
Yeah,
Lots of Army
I think that
personnel.
both the fact that EBV is in so many people that it's hard to find people who are negative and follow them, and MS is relatively rare, made this very difficult. But the way that they got to the statistical power they needed is just so impressive.
So this is a 20-year collaboration with the military.
Right.
They have a cohort of active duty U.S. military personnel from 93 to 2013 of over 10 million individuals. And all of these people are screened at the start of their service for HIV and every 2 years afterwards. And they keep serum. So they have 62 million archived serum samples from this stuff. In the
Your job.
They have the Department of Defense Serum Repository, DODSR. And so they got a hold of this, which is great.
Wow.
Then they could ask, what's the EBV status, and do these people have MS, and if they got it during the course of their service, and so forth. And about 5%.
Yeah, and go ahead.
5% of people were negative at the first sample. So of 10 million people, that's a lot of people, right? Hundreds of thousands of EB negative young adults just going into the military, some of whom could develop MS over time. Go ahead, Brian.
Yeah, and the first time I read this, I got to the 62 million serum samples part and sort of was a little bit in awe. I don't think that they actually tested all 62 million samples. They tested the first sample for all 10 million, which is pretty impressive to get that 5.3 number. But after that, they looked at medical records first of those 10 million to figure out who had MS. And then looked at those people's serum samples as well as some match controls. So I was trying to explain this paper to someone earlier, and I had it on my computer, and I said 62 million serum samples, and they immediately said, no, there's no way that's possible. There aren't enough authors on that list to measure 62 million serum samples. So they did not actually look at them all. That took me a little while to to work out.
So going through the records, they have 995 MS cases among these people. And so for each one, they got serum samples and they analyzed those. 801 MS cases and 1,566 controls with samples available to assess EBV infection status.
Yeah, so the 801 were out of the 955. It was sort of 801 who had samples they could use. So
actually, this is interesting. You in terms of the approach, which I maybe they detail it in the methods. I didn't look at the methods. It seems to me that you would start out by identifying MS cases, okay, right, and then try and find an equivalent number of EBV negative, MS negative people to power the study. And then you could probably quit your survey.
Well, so they actually they found a certain number of MS patients and they didn't look at their EBV status first. And then they found for each patient, they found two matched controls. And again, they didn't think about EBV status at this point. They just looked at MS patients and two matched controls per patient.
So only one of these 801 MS cases was in an EBV negative person. And we'll get back to that in a bit.
That is a person who both had MS and was EBV negative at the time.
Right? Seronegative, yeah.
At EBV at the time of the last blood draw, which could have been up to 2 years before that diagnosis.
That's right.
Okay, so at baseline, 35 MS cases and 107 controls were EBV negative. All but one of these 35 EBV negatives got infected during the follow-up with EBV, all seroconverted before the onset of MS. The median time from the first EBV positive sample to MS onset, 5 years. So they say the high seroconversion rate among individuals who develop MS during follow-up, 97%, is contrast with 57% rate of seroconversion among the people who did not develop MS. That's a 32-fold hazard ratio for getting MS after EBV infection.
And that's basically the bottom line here, I think, right?
It is.
The 32-fold hazard ratio.
Yeah.
With a really cool control.
Yeah,
32% hazard ratio based on seroconversion and a 26% based on diagnosis. Both of them are just...
Yes. So did these troops that became MS positive acquire their EB virus while they were in the service?
Yeah. Yes,
so that means that they caught it from other servicemen that they were serving with next to each other.
Yeah, sure. Or their family. Yeah, that's how
is that a suable offense?
No, because
you only really know they got it during the time after they started in the military and were measured. That you don't know if they got it at home or while they were in the field.
Yeah, they took leave
or on leave.
You're absolutely right.
Yeah,
there's lots of opportunities.
So they. As Rich said, they use cytomegalovirus as a control because it's like EBV, it's a herpes virus transmitted through saliva, similar racial, socioeconomic disparities in infection in the U.S. So the seroconversion for CMV, which they could get from their samples, same rate in those who got MS or not. So no difference. And in fact, the MS risk was lower among CMV positive people, which they say maybe the immune response attenuates EBV to some extent. They actually did a wet experiment here. I guess serology is wet too. They looked at serum concentrations of neurofilament light chain, which is a biomarker of neuroaxonal degradation. So it's not specific to MS, but it would be a marker in patients with MS, all right? Basically, it correlates beautifully with the onset of MS. No signs of neuroaxonal degeneration before EBV seroconversion. So EBV infection precedes symptom onset and also the first detectable pathological sign, which is this degradation of this protein. They also did a Vir scan where, if you remember this, we did this paper years ago. It's developed in Steve Elledge's group. You put the full proteome of all the known human pathogenic viruses on a chip. 200 different viruses, 110,000 peptides, 56 amino acid peptides with 28 amino acid overlap. And then you flow serum over it, and you can say, what antibodies do you have, right? The history of your infections. So they want to know what viral proteins are being detected in their serum samples. And they find a lot of EBV proteins, right?
Right.
But which you would expect. The overall response to peptides was similar in cases and controls, except for EBV. The cases, there was more of a response. But they say this argues against the second virus perhaps playing a major role in MS, because you don't see any specific peptides recognized by antibodies from the MS patients compared to the controls. And that's really it. They go through the possible confounding issues, but I don't think we need to go through that. They do talk about this one case that was EB negative, which, as Brian said, was gotten 3 months before onset of MS. But they say this person could have been infected after the last blood collection, right? Or maybe just didn't seroconvert, or maybe was misdiagnosed. They mentioned something called etiological diversity, right? Which means that a condition can be caused by different things, like poliomyelitis can be caused by other viruses, right? Not just poliovirus. So, extremely low MS risk in EB negative individuals suggests that by far most MS cases are caused by EBV, and thus could potentially be prevented by a vaccine. Wouldn't that be cool? Prevent MS with a vaccine?
That would be awesome.
Boy, I have a hard time thinking we're going to have a vaccine to prevent EBV infection population-wide.
Huh.
Yeah, because once you get MS, it's too late, right?
That's true.
Yeah.
And trying to imagine who would be your population to vaccinate, how young you would have to.
Well, and as you have said, Brianne, for HIV, it would have to be sterilizing, because a few viruses get in, they go latent, and that's it, right?
But I think we can discuss this further when we get into the mechanism. Okay,
right.
There may be opportunities here for very early diagnosis, and understanding the mechanism may help, inform treatments.
Are these congenital? And I know some of them are. The EB is a congenital infection also.
EBV?
Because, cytomegalo I know is, but...
For sure. No, I don't believe EBV is, no.
The other one is.
I don't think so.
CMV, yes, for sure.
Yeah.
Yeah.
They also talk about antivirals modifying the course of MS. But maybe when early onset, there's still some contribution of virus. Is that possible?
I guess maybe. It's not clear that the virus is still present and still causing the
this does
diagnosing many patients.
It does do a latent infection. Okay, and that latency probably involves a certain very low level of replication going on pretty much all the time. So I think there's opportunity here for continued insult by the virus.
But, from our previous paper, if you have polio, if you're paralyzed, giving you a antiviral is not going to help.
No.
Yes, Dixon, what were you going to say?
No, but if we could move on to the next paper, a lot of this clears up, because they've identified a protein that looks a little bit like a normal
the next paper is also fabulous. I just want to mention one more thing. So they point out that current therapy for MS is anti-CD20 monoclonal antibody. CD20 is a B-cell marker, right, Brianne?
Yes, it is.
And what I find interesting, and they a fraction of these patients who get these monoclonals, one of them is called Tisabri, they develop.
Well, Tisabri is not an anti-CD20.
What's Tisabri?
Tisabri is an anti-integrin. So you stop cells from trafficking.
Which a CAM is a trafficking protein, yes?
Yeah. So the anti-CD20 is rituximab.
Okay.
How do you
because because what was I going to say is that in a fraction of patients who take Tisabri, they get PML, progressive multifocal leukoencephalopathy, because they're immunosuppressed, right? So the latent JC virus, which we all have, starts to reproduce
in the brain
and causes. So is the integrin antibody also immunosuppressive?
So the integrin basically stops all T cells from going to the brain. So you can't get the T cells to go to the brain to cause pathology, but you also can't get T cells going to the brain to
knock out something.
Protect against JC.
So that is why you get JC reactivation or uncontrolled replication, right?
Yes.
Yeah.
JC, just to be sure, that's Jakob Creutzfeldt syndrome.
No, it's John Cunningham.
You see, that's confusion. I don't think you should use initials in this case.
Well,
I actually had a student give a presentation on polyomaviruses on Wednesday, and I learned it was John Cunningham on Wednesday.
Yeah, it's John Cunningham. And it's funny because I'm at the Tisabri website, and it says your risk of getting PML is higher if you have been infected with the John Cunningham virus. Interesting. Yeah,
polyoma, by the way, we don't have to go into this, but it's a completely different virus than the herpes viruses. But again, one of these things that infects just about everybody, and you just live with it. You never know it. Okay, except under certain circumstances.
Exactly.
Certain circumstances.
Right. So the antibody that's used for MS that they talk about is an anti-CD20, which is rituximab. And the idea with that antibody is that you're going to kill memory B cells because CD20 is on memory B cells.
But Tisabri is also used for MS. Yes,
yes.
In that case, you get reactivation. But so CD20 does not lead to PML?
No, because what CD20 is doing is it's killing the B cells in which the latent virus is.
And that improves the MS, right?
Right. And that seems to improve the MS. And there's sort of a question of how much that's because you're not making antibodies anymore from those B cells versus how much are you getting rid of the virus factories.
So an antiviral would kind of distinguish between those two, right?
Yes.
We must have some EBV antivirals, right, Rich? We have lots of herpes antivirals or some.
Boy, that's a good question. I don't know, because you would think that a lot of the antivirals for one herpes virus would be good for others, but that's not really the case. So I don't know about specific EBV. None come to mind.
So it says here, acyclovir and ganciclovir reduce shedding, but are ineffective clinically.
Okay.
Interesting. But there are some investigative drugs. So you could make one reasonably quickly now. All right. So let's do the second paper here.
Yes.
Which, where is it? This is a Nature accelerated preview. Clonally expanded B cells in multiple sclerosis bind EBV ABNA1, Epstein-Barr nuclear antigen 1, and Glial Cam. So a virus protein and a cell protein.
Great.
Tobias V. Lands and William Robinson, first and last authors.
So, Rich, this is where I got my idea about gene stealing, because how unlikely is it that one viral protein, which is a nuclear antigen, would resemble a glial cam molecule?
Yeah.
Well, all you really need is a peptide, right?
Yeah, that's
that
pretty
looks the same.
Yeah,
but that could have been like a gene that was altered and eventually evolved into the EBNA1, but it retained the antigen.
But it doesn't have to be, though. Peptides are pretty short, and they can be found.
Okay.
So
you only got 20 choices.
It's just my mind running amok.
So the first paper we see an epidemiological link. Now they say here, there's some suggestion that molecular mimicry is involved. In other words, You make an antibody against a viral protein, and just by chance, it can react with
Right.
the cell protein. But it's really hard without the current tech that we have today, which is amazing. You couldn't really figure it out.
Oh no! When I look at the technology that's involved in both of these papers, it's amazing to think about. How dependent these discoveries are on this just incredible technology.
Yeah, molecular mimicry, we've been able to kind of hypothesize about it and sometimes kind of show some limited peptide overlap, but the tech is really what makes this go further than our past.
As an introduction to this, Brianne, maybe you could do this sort of overview, this idea of what I would call, I'm making this up, pathogen-induced autoimmunity.
Okay. So what should normally happen is that you have B cells and T cells that are made with a huge diversity of receptors. They bind to self proteins as well as non-self proteins. And then you have a process where you get rid of all the self-reactivity. So in the end, you should really just have non-self-reactive B cells and T cells out patrolling your body. And there are that'll happen before the cell is allowed out into the body. So we're all doing this right now in generating new cells, but we don't let them out until we test them. And we also have some mechanisms that turn off any self-reactive cells. So these are all called the tolerance mechanisms. Right. And so what has to happen with any kind of autoimmune disease is that somehow you have to turn on a cell that should have been turned off or that was otherwise turned off. You have to get through this tolerance crack.
Why don't we just kill those cells?
And there, so in some cases we do kill those cells.
What good are they?
In some cases
What good are they hanging around? Because they can cause problems later on in our lives.
So there are a couple reasons why we don't kill them. The simple reason that I can tell you now is that some of them, the B cells, for example, can't actually check to see whether they're self-reactive against everything in the body during development. All proteins are not available in the bone marrow. So we have to have some other ways that they can be restrained when they see a self-protein outside the bone marrow.
Okay.
And so what has to happen is that somehow infection can mess up that tolerance process or otherwise break through that tolerance process. Sometimes you might, in the case of molecular mimicry, turn on a B cell with a pathogen because of the inflammation that comes with that pathogen. And then it just so happens that B cell recognizes some amino acids that are also on a self protein.
So it'd be cross-reactivity between something in the pathogen and some self protein.
Exactly. The self protein never would have turned on that B cell. But when you have the pathogen and the inflammation around, you get that B cell.
So there are some modern chemotherapeutic approaches to treating cancers where they use monoclonal antibodies to uncheck the checkpoints that these T cells were held in check for, so they can go out and kill the cancer cells. But I'm told that in 20% of those patients receiving those kinds of treatments, it reactivates some T cells which attack normal tissue. And so those are the cells that we wish wouldn't be activated. But they said 20% of people receiving that kind of therapy go on to develop some kind of an autoimmune disease.
Yeah. So do you remember how I said that we have a bunch of ways that we kind of regulate or keep those cells turned off when they're out in the rest of the body? It turns out those are pretty important. And a lot of the cells are getting turned off by those mechanisms. The checkpoint mechanisms are exactly those mechanisms, or at least are some of them. So this is happening in everybody.
All right. So in this paper, they have MS patients. They get cerebrospinal fluid, and they sort single B cells from the cerebrospinal fluid. And they also have a non-MS patient cohort. These people have neuroinflammatory diseases, but not MS. So they sort from both, and they clone out the paired heavy chain and light chain sequences to get the antibody. It's kind of like what Amy was talking about last week. And so they got lots of paired sequences from blood and from CSF B cells, okay? They find that, and this is where I would like Brianne to explain, that the repertoire from these patients is highly clonal, suggesting antigen-specific proliferation of select clones within the CSF, and they don't see this in the control patients. So what does that mean?
So what that means is that when they look at the cells that are coming out of the CSF and they sequence their heavy and light chain genes, they are largely seeing cells that have used the same mutations and the same combinations to get that heavy and light chain gene. So they must have come from the same original precursor cell that had mutated and then expanded. It's less likely that you would have gotten all of those same genetic changes coming from a bunch of different precursor cells. So these are all progeny of the same original B cell that made those mutations.
Specifically in the cerebrospinal fluid.
Yeah, so that might mean that one B cell made it into the cerebrospinal fluid, and when it got there, it made a lot of progeny.
Are we supposed to have B cells in the cerebrospinal fluid, or is that a boo-boo?
I think that it's probably okay. So the reason why I'm hedging on that is that if you had asked me as a graduate student before the Tisabri thing happened, if you would ask any immunologist before the Tisabri situation, we would have said, well, you're never supposed to have T cells in the brain. And then we blocked T cells going to the brain and everybody got PML, or some people got PML, because it turns out those T cells were really important for protecting you against JC. So I can't say that you shouldn't have
So the
T
notion
cells.
that the central nervous system is very tightly immune privileged is maybe it's a little leakier than that.
It's maybe not keeping all the cells out. Maybe it's just telling the cells to stay really quiet. Maybe it's more about the environment and the cytokines that are being produced than actually being a whole wall.
I have another question about this compartment later on, but it's not relevant yet.
Okay.
They also do the sequence of these antibody genes, and they say from the sequence they can tell that there has been somatic hypermutation going on in the CSF, right? Suggesting ongoing intrathecal SHM.
So that came around quicker than I thought. Because that's my question. Because of our previous discussions, I think of somatic hypermutation as going on in germinal centers. Okay, which implies lymphoid tissue. This is happening in the cerebrospinal fluid. How am I getting somatic hypermutation in the cerebrospinal fluid?
I don't have a great answer for you. But I can tell you that that when we think of germinal centers, we often think of, the lymph node or the spleen, but there are some kind of lymphoid tissues in other places that are smaller. Pyrus patches, but also, tonsils and adenoids and sort of specialized tissues in a few different organs. And I don't know enough about the spine to know if there are any specialized tissues there or not.
So they tell us that in general, MS patients have high immunoglobulins in their CSF, and it's oligoclonal, as we have just said. So they isolate immunoglobulins from CSF of their patients. They do mass spec to characterize them, and they compare those with the sequences that they've just got from the individual B cells. And they say for 87% of the clonal sequences, peptides matching the variable chain sequences unique to the patient were identified, but only 40% of singleton sequences. So they say that highly expanded plasmablasts are the predominant source of this oligoclonal B cells in the CSF.
So a smaller fraction of the B cells are making most of the antibodies.
Right.
And they're all IgG based, not IgM. That's another question, of course.
Most likely, I don't know that I'm not sure that they've measured that, but most likely, yes.
The IgM would have a very difficult time getting into the brain from there, wouldn't it?
So next, they made these antibodies from the genes. They made 148, and they produced monoclonal antibodies, and they asked, what do they bind? So they have protein microarrays with EBV proteins. And 7 other MS-associated viruses, by the way: measles virus, rubella virus, and varicella-zoster virus.
Cytomegalovirus as well. HSV1, HSV2.
That's in that list?
Well, I'm just looking at this chart of the peptide array. I don't know about their list,
maybe
but I'm seeing.
I think maybe that's not an MS associate. Maybe it's just a control.
Yeah.
Well, no, they say 7 other MS-associated viruses, but then the text
they
only
use 4.
lists 3.
Yeah. That's 1, 2, 3. That's right. Okay. What are the others besides herpes?
I'm seeing on this figure, I forget, I don't know, what figure is it? Figure 2A, I'm looking at their peptide arrays, and I'm looking over at the viruses that are represented, and I'm seeing measles, rubella, varicella-zoster, CMV, HSV1, HSV2, and a HER-W, whatever that is.
All right.
Oh, is that the human endogenous
retrovirus?
Yeah.
Interesting. All right. 1/3 of the MABs bind EBV proteins and peptides, and 20% bind to other viruses, the VZV and CMV. Half of the VZV antibodies cross-react with CMV and EBV.
Because they're all herpes viruses and they have common epitopes.
Yeah. Monoclonals from 6 out of 9 MS patients bind a transcription factor of EBV called EBNA1. And antibodies binding peptides derived from EBNA1 were found in 8 of 9 patients. And a specific region of the EBNA, amino acids 365 to 425, is the target of strong antibody responses. And their peptide and protein microarrays showed that this particular monoclonal, MS39P2W174, binds EBNA1 within this region of amino acids 386 to 405.
Recall that EBNA1 popped up in the previous paper as well when they did
that's right.
Peptide arrays for antibodies from the MS patients. That was the most prominent hit.
And they did alanine scanning mutagenesis, where you change every residue to an alanine in a protein and ask, does that muck up its function? They say the function in this case binding to the antibody. They say, yeah, this is the region that's a central epitope for this antibody, this AA394 to 399.
So EB replicates in the nucleus?
Yep.
Yes.
Yep.
Thank you.
Yep, it does. All right. Then they do the crystal structure of EBNA 386 to 405, the epitope, together with this monoclonal with the long name, the name that shall not be named. Do high resolution structure so they can see exactly how the antibody and the peptide is interacting.
So you want to go out for a beer? No, I got to do this crystal structure. Maybe later.
You could have cryo-EM. You could do cryo-EM and have your lunch also.
So this particular monoclonal, the gene encoding it is one of the Ig heavy chains overrepresented in the CSF. And they say all but 2 of the residues that interact with EBNA1 are unmutated germline residues. You like that, Brianne?
I actually kind of don't like that, because that means that many people are likely to make this antibody.
Right. That's right. Because they're probably in most of us, or all of us, right? Yeah.
Yeah. Many, we all have that. We didn't have to have the luck of making those.
So they say, maybe the unmutated ancestor of this antibody, germline, right, without any changes, maybe that had a propensity to bind EBNA, as Brianne spoke earlier before. So they actually do some experiments which show that You don't need the somatic hypermutation to get EBNA binding. Naive B cells have EBNA1 specificity. That's what you would expect, right, Brianne?
Yes, exactly.
But there's a somatic hypermutation event that is peculiar in maturing this reactivity for the glial cam.
Yeah, that's right. Yes, we're going to get to
exactly.
that. Right. All right, so then they do human protein array. So now we want this antibodies in the CSF are binding EBNA1. Fine. What else do they bind? So they throw human proteins on these microarrays, 16,000 proteins, which is 80% of the proteome. This is just amazing that you can do this, right?
Yeah.
And so this antibody with the long name binds glial cell adhesion molecule, Glial Cam. It's an Ig superfamily adhesion molecule. It's made in the CNS by astrocytes and oligodendrocytes. I mean, what do you think they've said when they found that, right? Because
probably words that
that's
we
right.
don't want to say.
This is a family show.
Listening.
One of the hallmarks of MS are these plaques in the brain, and this protein is in them. Glial Cam is in those, they call chronic active plaques. They do a lot of experiments here. And one of them that I like, they stain mouse brain with this antibody. And you can see what they call predominantly glial patterns in the mouse brain.
So could somebody please do a little brain cell biology for me? I missed that lecture. Okay. And so glial cells, astrocytes, oligodendrocytes, how do they all fit together in the brain? Well, who does what?
They wish they knew. Rich, that's what they're working on. Nobody knows the exact architecture of the brain.
Glia are kind of, Brianne, are glia macrophage-like?
One type of glia are macrophage-like. So I think of glia as sort of the cells that are supporting neurons. Yeah. Again, I'm not an expert in this, so I thought glia could include astrocytes and oligodendrocytes.
Yeah, okay.
But I'm not positive.
So astrocytes are a major cell type which are supportive, right? But they're also a defensive. They're a defensive cell. When you get an insult, they react by producing things that are supposed to help but may damage neurons.
And there is one cell which facilitates myelin sheath formation.
I think that's
oligodendrocytes.
That's
right.
So we got neurons, and then we got all these other support cells that help keep the neurons in good shape.
Here.
Yeah, remember the disease is mainly loss of the myelin sheath around the nerves, right?
But that doesn't have to be direct. We could mess up one of these support cells, and that could indirectly result in loss of myelin sheath. Is that a fair statement? Because this is where I'm going with this. Because I'm thinking of the mouse model where we and we'll get to this, where we inject mice with myelin basic protein, which is a component of the myelin sheath, and generate an autoimmune response to that. So you're attacking the myelin sheath directly. But in this case, we're talking about a protein that's in a support cell, not in the neurons.
Yeah, wasn't that the original?
So
negative aspect of Pasteur's rabies vaccine. He was using the rabbit spinal cord as the source.
There was a side effect that was I was just reading about this.
Right.
Yes.
Rich, oligodendrocytes are the myelin-producing cells.
Oh, okay.
Yeah.
So
and I think one of the so people use myelin basic protein, like you mentioned, in MS models. One of the other proteins that they sometimes use is called MOG, or myelin oligodendrocyte glycoprotein. And I would bet that comes from the oligodendrocyte, which makes me think that if you attack the oligodendrocyte, you have a problem.
Okay. So you can attack the myelin sheath directly, or you can attack cells that are responsible for making or maintaining a myelin sheath, and the downstream, the result's the same.
Right.
Yeah.
But if you attack the astrocytes, you destroy Houston.
All right. So this antibody, MS39, as we said, the one they pulled out of patients and the unmutated germline ancestor, they both bind EBNA1 similarly. But bind glial cam with higher affinity, all right, 10-fold higher affinity. So why is this? They noticed that the antibody epitope is in the cytoplasmic domain of glial cam, and they know that this part of the protein happens to be phosphorylated, and their structure of the antibody peptide epitope suggests that phosphorylation could have an impact, and indeed, phosphorylation increases the affinity like 50-fold, and it makes the structure really helps them here because they can see exactly what the phosphorylation is different. So basically, they say post-translational phosphorylation enables this antibody to cross-react. So it's an EBNA antibody. The cell protein is phosphorylated. Now it recognizes glial cam really well. Did I get that right, Brianne?
You got that right.
Isn't that amazing?
Yes.
And as I say later, some kinases are risk factors for MS, right? The kinases that put these phosphates on.
Ah.
Isn't that something?
Mm-hmm.
All right. What else should we talk about? All right. So is anti-glial cam reactivity a broader phenomenon in MS? They have 147 other antibodies that they had cloned out. So they asked, do they react against glial cam? They found 10 additional MABs from 7 patients that bind glial cam, and then 7 more that bind the extracellular domain. So you get these patients are making antibodies against multiple glial cam epitopes. All MS patients and most healthy individuals have plasma reactivity to EBNA, right? You get infected with Epstein-Barr, you make antibodies to EBNA. But the patients have increased reactivity to glial cam. And so I guess they never really tell us why, right?
Well, part of it is this somatic.
I can hypothesize why.
The somatic hypermutation, right? The idea is that in the people who develop the disease, the original EBNA reacting B cells
they get in the brain.
undergo somatic hypermutation, apparently in the CSF.
Yeah.
If I understand it correctly, and mature to a form that cross-reacts with glial cam. Is that?
Yes. So yes, there's one more piece to it, though. So we could say, okay, everyone has antibodies against EBNA1 from these naive B cells that are at the germline, in germline configuration. We all, get EBV infection. We all make these antibodies and we all stimulate these B cells. Mm-hmm. But the only way that those B cells that happen to bind to this particular epitope of EBNA1 are going to get to do somatic hypermutation is if they get some T cell help. And that has to be a T cell presenting glial cam 1. Each of the 4 of us, are going to present a different part of glial cam 1 because we each have different MHC types. And so only one of us perhaps has the right T cell to help this B cell make this antibody and to let this B cell do its somatic hypermutation. So the one thing that is missing here is, in theory, you could look at the HLA types of the people and see which HLA type is associated with presenting the glial cam antigen. And I think that would be kind of... I think
they mentioned that there is some association of haplotype, right, with MS. Yeah.
That's the one thing that you can always see with most autoimmune diseases.
It's just a crapshoot, basically.
Yep.
Some people are just going to get MS and others not because of polymorphism of HLA.
So some people are going to get EBV. So you have to get EBV, you have to have the right HLA, and the right B cell and T cell have to get stimulated during the time when you have EBV.
So the first part, you have to get EBV. That's common. Most people get EBV at some point. But what's rarer is the HLA type and the right T cell, right? Wow.
Right, exactly. So, each one of those kind of is a funnel.
That's incredible.
There is one more experiment here. Rich mentioned it briefly, but there's a mouse model of MS. It's called experimental autoimmune encephalomyelitis. You immunize, you give the mice. What do you immunize them to do this? I forgot.
In this case, they gave them PLP.
PLP, right?
Which is proteolipid protein. There are 3 different things you could give them.
So they immunize the mice with this ABNA1 peptide that we've been talking about, and then they give them PLP. And so they say the ABNA group, they get antibodies to both ABNA and Glial Cam. They got a strong T-cell, CD4 T-cell response. And the peptide stimulates cytokines from these B cells. And the ABNA group shows more paresis and CNS immune cell infiltration and demyelination. So kind of providing more support for this.
Basically
idea.
Exacerbates the model.
Yeah.
Adding ABNA protein.
Yep.
ABNA peptide.
They also pulled PBMCs from MS patients. They stimulated them with ABNA and Glial Cam proteins and compared them to PBMCs from healthy people. And I guess they show the higher cytokine expression with the ABNA Glial Cam from MS patients compared to healthy individuals.
Right. So it looks like the ABNA specific T cells are activated in those people.
So that's the story here. It looks to me. I mean, for years I've heard this story, but I never believed it because the data weren't there. But these 2 papers today, I think, are really convincing, right?
So then, what do you do?
Hold on, let me see. So we demonstrate the presence of cross-reactive antibodies in 20 to 25% of MS patients. So is it because we just didn't find them in the others, or is there something else going on in those others?
Maybe it's something else. Maybe it has to do with, say, another part of the immune response, or maybe this goes back to our first paper about timing of when you're seeing some of these responses and when you actually are looking in the patient.
And they also mentioned several kinases have been risk genes, MER-TK, MAPK1, and 3, TYK2, potentially contributing to phosphorylation patterns. That's interesting. So they must, these kinases must differ in different people, and so that gives you a risk factor, right?
So does any of this that you've
so.
heard so far suggest intervention strategies?
Well, that was the idea in the previous paper, either vaccination or antivirals, right?
Yeah, well, so far not so good.
Because as Brienne said, if ongoing reactivation contributes to the progression of MS, right?
Right.
I don't know, but once you have the antibodies, Brienne, I'm not sure what would happen, right? Once you have the cross-reactive Glial Cam antibodies,
Yeah, you don't
I guess
need the
maybe
virus anymore.
Right.
maybe the virus would induce more synthesis of the antibodies, right?
Yeah, maybe it would trigger the B cell to make more antibody. But I think that's really the question is, is the problem that you have to prevent making antibodies in the first place, which might happen with a vaccine, or is the virus itself actually causing pathology? And so I think there's sort of evidence that could go either way. That anti-CD20, like we said, it could be killing the cell that's making the virus, the B cell, or it could be killing the cell that's making the antibody, the B cell.
Yeah. But what do you do? What do you do to sort this out? You're not going to make a vaccine or an antiviral to answer this question, right?
So I'm thinking,
right.
I'm looking ahead to the era of future modern personalized genomic-driven medicine, where your genome and its analysis are a fundamental part of your medical record. Mm-hmm. And we understand this well enough to understand whether your genetic profile puts you at risk for this because of these various things like HLA type and kinases and everything else. And in that case, knowing the mechanism, perhaps there can be therapies designed to prevent you from developing this. I'm really wigging out here.
Brianne, could you CRISPR out these particular antibody genes?
Probably not, because they probably respond to other things when you combine them slightly differently, and you wouldn't want to lose that reactivity. I think that we've got to think about, do we imagine whether we need that antiviral or vaccine? I'm sure that many of my college students who really don't enjoy suffering from mono would be on board.
Absolutely.
But I guess I think those are the only
what
ways
is
we
the
can really figure this
what
out.
is the average age for MS onset? Do we know?
It's 30s.
There's 3 different kinds of MS, and I think they vary, but I would say 30s or 40s.
So you could immunize 20-year-olds, right?
When do people first get their first infection?
Some of them do. Some of them get them young, but a certain fraction of kids don't get them until they go to college, right?
That's where mono comes from, yeah.
Then the rest of them get infected in college.
So actually, this raises an interesting question, because there's as I understand it, at least, there's a pretty strong correlation between a delayed encounter with EBV and getting mononucleosis, right? The people who get mono are the ones who, among other things, didn't pick up EBV earlier in their lives. I wonder if there's any such association with MS.
Yeah, if you could look at these people and then say, did you have mono?
The other question I would have is, can you use CRISPR to attack the virus itself and get rid of the EBNA1? Just knock out the virus like that.
So the virus is latent in memory B cells, long-lived memory B cells.
Yep.
It's episomal in the nucleus.
Right.
So you'd have to get every B cell. I'm not sure it's possible. Yeah.
Just a random question.
No, it's a
no. I mean, these
good
are
idea.
Good idea. I mean, people are trying to figure out whether you can use techniques like that to get rid of your endogenous HIV, right? Cure HIV the same way.
A monospecific FAB fragment to block the epitope on the myelin sheath to prevent the damage from the divalent antibody.
Yeah.
How's that, Brianne?
That could stop the further damage.
That's the point, because these people are diagnosed pretty early on, and they know they have MS before it's symptomatically inhibiting for their daily lives. They know they have it, and they know they're going to go on to progress into it, but they live in anticipation of a bad future. I know several people with it.
Yeah.
And so you could stop it there and prevent that from happening.
Yeah.
That could be especially true if you could come up with a test to look and see if people are making this specific
That is exactly
antibody.
Right.
So if you could say, oh, this person's making that antibody, they're likely to progress to
so
MS
with familial,
later. You could do some interventions.
You mentioned haplotypes too. That suggests a familial trail. Could you go back in their family history and find people that identified with that haplotype? Obviously, you inherited it. So MS, is it inheritable?
Maybe, yeah. Well, no, because you have to have those. It would yes and no, because
propensity, yes,
the HLA is inheritable.
No. That's right.
But then you still have to get EBV, and you still have to be the unlucky person that has the wrong B cell, get the wrong stimulus.
That's right.
So look at the tech that went into this. We have these proteomic scans of antibodies, and there were more that they did. We didn't talk about. They did phage display. They did viroscan. We have crystallography. We have our understanding of antibody structure, single cell sorting. We have
and 62 million serum samples.
And then the serum sample, that incredible resource.
All these peptide chips.
Yeah.
Well, maybe you already did that phage display.
Yeah, I mean, it's just all coming together. And there are not that many authors on the 2 papers combined. It's really impressive. So this, I think, will, as we have just been chatting here, what is the word? We've been bantering.
bantering.
People are going to think about how to prevent this because it's a crappy disease. And if a vaccine could prevent it, hell, why not?
That's right.
Oh.
One vaccine prevents mono, Burkitt's, MS.
Right.
Yeah.
So MS is not considered a hereditary disease. However, this is wiki, of course. However, a number of genetic variations have been shown to increase the risk. Okay.
Well, that
and I'm looking at it. Looks like HLA genes are prominent in that mix.
Sure.
Well, no, it doesn't contradict because you would say that it was hereditary if you could always identify parents and children who had it. Well, and since you have to have the gene and other events, it wouldn't.
It's not real obvious, right?
Something you could do.
So
in
everything
a pedigree,
that looks random on the outside probably isn't. We just haven't found the mechanism yet. So all of these other things that you were discussing that need to happen quite a bit because there's a whole bunch of MS patients out there. It can't be that random. It's got to be another underlying genetic basis for it. That's my own opinion, by the way. But
you're right. It's not random, but it's a toss of the genetic dice, basically.
It's an unlucky sudden combination of all these things at once. That's right.
I mean, life is not perfect.
No, well,
and the things that make it cool, the very things that make it amazing, can kill you, right?
Well, yes. And eventually, let's face it, I don't know anybody that's actually outlasted everybody else, but I think we're all heading for the recycling bin.
Well, nothing lives forever, Dixon.
No, that's the point. That is
except vampires, right?
I guess.
And they say they don't really want to live forever. It's too hard to get blood.
Choice. You see, that's a genetics and no, we don't.
All right, folks, that's enough of that. Cool papers.
Yes.
Let's do some picks. Dixon, what do you have for us?
Okay, let me jump down here, and I will explain my pick very simply by saying that we now have discovered the history of the drought that's affecting the North American continent, and particularly the southwest of the United States. We have now a 1,200-year-old history accumulated by looking back at tree rings from 800 CE, whatever that stands for. I used
Christian
to
era. No, it's not Christian era.
No, CE.
Is it Common era?
Common era to today. And they have found these authors, 3 of them, have found 2 mega droughts. One was in 1500, and the other one was last year. And they predict that this is a short communication in nature. They predict that this drought will continue on for some time. And this is really mucking up food production, hydroelectric power generation, obviously recreational things. Whatever is to do with water is screwing everything up. And if you want to know what happened to the Anasazi Indians, or the Native Americans, or the first peoples, however you'd like to refer to them, there's evidence that the reason why they abandoned their 2,000-year-old site was because of this drought. And you look at the tree rings on the ladders leading up to their places where they used to sleep, and they're almost, you need a microscope in order to see the difference between the rings at the end of their construction to show how severe this drought was and to what extent these people went to produce irrigation schemes that kept those societies intact for as long as they could, and they eventually gave out. Well, guess what? We're stuck with the same problem today. The rivers that supply the irrigation water for all of those states, Nevada, Arizona, New Mexico, and California, come out of the Rocky Mountains of Colorado. And this is a big issue that's going to get worse and worse and worse, apparently. And we can trace it right back to this phenomenon that we're suffering from.
Dixon, was the 800 CE drought, that was not anthropogenic, right?
No.
I wouldn't think so.
No, not at all. Not even the 1500. But the one that's occurring now exacerbates it. Let's just put it that way. Like the hyper J and D regions of the antibodies that exacerbated the MS. This is the same, only on a different scale. This is depressing because you're a victim of the weather and climate, and these two things converge together to produce some really awful situations right now.
It's our fault. It's our fault.
Well, we're prolonging it.
Yeah,
we're prolonging it. That's the point. There's no groundwater left out there. They're fighting over the water that comes out of the rivers. There are literally thousands of dams along the Colorado that are being controlled by people that are not releasing water further downstream, and the state of Colorado has an agreement with all the other states as to how much water they will release to each of them, and that all has to be changed. Imagine the hydroelectric dams that don't have enough water behind them to supply enough pressure to generate enough electricity to keep places like Las Vegas going. Not that everybody cares about that, but 700,000 people depend on electricity generated by the dams along the Colorado River.
Dixon, we have a lot of seawater. Can't we use that?
For what?
Figure it out. It's tons and tons and tons.
Well,
of seawater.
We have to learn how to make it into fresh water. Cheap.
Can't we be clever and figure it out?
Cheap. We figured it out. We know how to do it. There's a lot
it's
of
expensive, right?
It's very expensive. It's very expensive.
Why? Why is it expensive?
Because the technology is expensive. They're using reverse osmosis.
I have an idea. Get Elon Musk to figure it
out. Excuse me for laughing, but I've really immersed myself in this subject over the recent past, and it looks intractable. It looks almost like no cure for MS, no cure for the American Southwest.
Dixon, this is not an uplifting pick.
No. Sorry about that.
It's okay.
No, I thought that Riches was the most uplifting pick. I couldn't stop watching it, but they wouldn't start. The guy that's sitting below the woman, we won't get to that. Come on.
Brian, what do you have for us?
I have an online quiz that I took earlier this week called "Which Immune Cell Are You?" I saw it going around a few places in social media, and there are a number of questions where you have to say what you would do, and then in the end, you find out which immune cell you are. I will apologize to many of the other TWiVsters that at one point it does ask you what your favorite lab technique is, and they are very immunology-heavy lab techniques. There is no plaque assay. But I have taken this quiz, and it turns out that I am apparently a memory deep.
Is that right?
No
Good
kidding.
for you.
Yes.
I just took it while Dixon was going on and on. And I'm a natural killer cell.
Oh, dear.
I'm still taking it. What best describes you? How the hell do I know what describes me?
Says I'm spontaneous and know how to get things done.
No, I can't pick one of these things.
Vincent, you're grumpy. Where's the grumpiest immune cell?
Well, there's no grumpy. All right, forget it. We'll take it. That's very cool. Rich, what do you have for us?
I have. This actually I got from a Facebook post from McFadden's daughter, Shauna. So shout out to Shauna McFadden. It's Nikola Tesla 3D anamorphosis.
Right.
Okay. It's an artwork by a guy named Patrick Prosko that's in the Illusion Art Museum in Prague. It's from 2015. So given the structure of this thing, I don't know if it's going to be there anymore. I tried to go to the source. This is one person's take on this. And in fact, this particular video is better than even the official video. And it's one of these things where a collection of stuff looked at from a certain perspective looks like a portrait. Exactly. So when it starts off, you see Nikola Tesla, and then the camera moves around and you see that it's a display made up of all sorts of electronic bits and pieces. Okay. But then you go back to the original perspective and you see Nikola Tesla again. Who knows how they do this stuff? It's amazing. But I thought it was really cool.
That is cool.
I'm looking at the video below it.
There's an artist. I'm blanking on his name. Of course, he just died recently, and he was wheelchair ridden. But he painted enormous portraits, but they were all pixelated. If you got up close to them, you could not see the picture. You had to go far away in order to actually see this picture. This guy had the ability to be up close and see the picture. So there are people with that kind of perception, Rich, that can do these constructions, but they're amazing. That was amazing.
It looks to me like the Illusion Art Museum would be an interesting place to visit.
Chuck Close. The guy's name is Chuck Close.
Chuck Close. Okay.
All right. My pick is a new book by the team in Mexico. Susana Lopez, Selene Zarate, Martha Yocopicho, and illustrations by Eva Lobaton. They're all virologists, except for, I think Eva's an illustrator. This one is Paul's Uncle Doesn't Want to Get Vaccinated. And so they have produced these are children's books. They're free. They have made Paul Has Measles, Paul Stays Home, Paul and the Mosquitoes.
Right.
And now Paul's
singing group.
Uncle Doesn't Want to Get Vaccinated. Illustrated book for children about vaccines and vaccine hesitancy. So it's a free PDF, and you can download it or any of the previous ones. This one is available in Spanish and English, and some of the others are in many, many different languages. But you could find links in this blog post, which is on this blog that Rich mentioned before, Virology blog. We have 3 listener picks. First one is Bob, who sends a YouTube channel of a YouTube video of the 1969 Apollo guidance computer. This would be a great pick for listeners. It matches TWiV's meaty and rewarding dive into details, all told around a knuckle-biting moment in history. That's what we do, meaty and rewarding dives, don't we? I like that.
Yes.
Then we have Ann writes, a few episodes ago, you shared a listener pick for Decoding the Gurus, where the 2 hosts went through the Joe Rogan podcast episode with Robert Malone, discussing the misinformation presented on the episode almost point by point. It was a long episode, but worth listening to, in my opinion. That episode provided numerous links to sources of information, and one of them was another podcast called Debunking the Funk with Dr. Dan Wilson, a microbiologist. Turns out Wilson regularly posts somewhat shorter videos debunking anti-vax misinformation. He was a guest on this week's episode of Decoding the Gurus, and in it, he and the other podcast hosts of DTG expressed a lot of love and appreciation for TWiV. The episode description mentions this, but if you want to hear them discuss their high regard of TWiV, they start at about minute 29. This episode is only an hour long. They mention other podcasts devoted to debunking scientific misinformation too. She provides a link for that. As a longtime listener, I know that TWiV provides a lot of value, so thank you and the whole crew again. But I appreciate that folks are using your example and podcast to help spread information about science and how the scientific process works. And then Beth writes, I love your podcast. I've been a regular listener since a few weeks into the start of the pandemic. Consequently, I have become quite a critical listener to reports about COVID-19 in the popular media. Today, I was happy to hear a reference to TWiV and NPR's Morning Edition. She gives a link to that at about 5:25 into the recording. Inspired by Vincent's recent picks of cooking shows, I have one to recommend called French Guy Cooking on YouTube. Here's a link. Have you ever seen another kitchen with his mix of tools? But don't worry, he has plenty of videos for Italian cooking. Cool. Great picks. That'll do it for TWiV 869. Microbe.tv for the show notes, your questions, comments, picks, TWiV at microbe.tv. And if you like what we do, consider supporting us, microbe.tv/contribute. Your not deductions, your contributions are tax deductible. Why do I always say your deductions are tax deductible? Dixon Despommiers at Racaniello.org, thelivingriver.org. Thanks, Dixon.
I think I'll have another cup of coffee and come back to the clutch. This was absolutely fun. This was perfect.
I'm glad you like it.
Loved it.
Brianne Barker's at Drew University, BioProf Barker on Twitter. Thanks, Brianne.
Thanks. It was great to be here. I was so excited to read those papers. And now after this episode, I've almost figured out where to look on this new video setup.
That's funny.
That's right.
You'll get there. No problem. Yeah, it's a matter of getting used to. Rich Condit is an emeritus professor, University of Florida, Gainesville. He's currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time.
And I am Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology and the American Society for Microbiology for their support of TWiV and Ronald Jenkins for the music. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.